Welcome back, fellow researchers, to this explainer on the profound biological significance of 2-O-RNA methylation and the state-of-the-art sequencing methods we're using to actually study it. Now, as senior researchers, we all know the RNA landscape is way more complex than just a simple string of A, C, G, and U nucleotides. For decades, the epitranscriptome has been this fascinating hidden dark matter of RNA biology, right? It's heavily decorated with modifications that essentially dictate structural integrity and function. So today, we're going to shine a spotlight on one of the most critical, yet historically elusive modifications in that hidden layer.
So, here is our roadmap for today. We'll start by unlocking 2' O-RNA methylation, then look at stability and translation efficiency, dive into viral immune evasion mechanisms, and finally wrap up with the CD Genomics sequencing service.
All right, part 1, unlocking 2' O-RNA methylation. Let's get right into the chemistry to understand the biology behind this highly regulated modification.
At its core, 2' O methylation, or Nm for short, is a post-transcriptional modification catalyzed by specific methyltransferases. It's exactly what it sounds like, the precise substitution of a hydrogen atom on the 2-hydroxyl group with a methyl group, right at the 2' position of the RNA ribose ring. Now, you might be wondering, why does this matter so much chemically? Well, that original 2' hydroxyl group makes standard RNA incredibly susceptible to nucleophilic attack and base-catalyzed hydrolysis. By swapping it out, this ubiquitous chemical modification effectively neutralizes that structural vulnerability. And it's highly conserved evolutionarily, popping up across mammals, yeast, plants, and viruses. It basically acts like a chemical lamination, providing this absolutely indispensable protective seal for mRNA, tRNA, and RNA alike.
Moving right along to Section 2, Stability and Translation Efficiency. Let's see how this biochemical tweak actually translates into physical armor and regulatory control.
Think about the stark contrast between vulnerable standard RNA and shielded methylated RNA. Standard unmodified RNA is structurally flexible, but let's face it, highly vulnerable to degradation by ribonucleases in the cellular environment. But the moment we introduce that methyl group, we fundamentally alter the molecule's chemical properties. The enhanced hydrophobicity from the methyl group drastically boosts its structural stability. It essentially forces the ribose into a specific puckered conformation. This makes the RNA highly resistant to enzymatic cleavage and preserves the correct three-dimensional conformation, which is, of course, absolutely vital for proper ribosome assembly and function.
But you know, the story goes way beyond just resisting degradation. These modifications also function as highly precise sorting tags. In ribosomal RNA, this methylation is crucial for translation accuracy. It introduces steric hindrance that actively prevents unfavorable interactions and incorrect folding, minimizing errors during protein synthesis. And over in transfer RNA, it tightly regulates tRNA folding and maturation, ensuring that precise anticodon mRNA pairing actually happens at the ribosome. Honestly, without this chemical lamination, the basic mechanics of cellular growth and stress response would just begin to break down due to translational noise.
Okay, let's jump into Section 3, Viral Immune Evasion Mechanisms. We're going to examine how this operates in non-clinical pathology through a really compelling in vitro case study with HIV-1.
This sequence brilliantly illustrates what is essentially a molecular stealth mission. Researchers discovered in vitro that HIV-1 virus particles actually use 2' O methylation to hide from the host's innate immune system. Basically, post-infection, the virus actively hijacks the host cell's FTSJ3 methyltransferase. It forces the host cell's own cellular machinery to add 2' O methylation modifications to its viral RNA. And because the viral RNA is now cloaked in these host-like methyl groups, it completely bypasses detection by MDA5, which is the cell's primary cytoplasmic RNA sensor. Just like that, the virus successfully and silently evades immune recognition.
When researchers compared wild-type cells against cells where that FTSJ3 enzyme was knocked down, the results were incredibly striking. By knocking down FTSJ3 in infected cells, the virus simply couldn't methylate its RNA. The result? MDA5 recognized the naked viral RNA right away, and they observed a massive spike in type 1 antiviral immune response signaling. We're talking huge increases in interferon alpha and beta expression. This elegantly proves that this specific RNA methylation is the exact non-clinical mechanism enabling viral replication and evasion here.
Now, historically, identifying these hidden modifications was incredibly time-consuming, and researchers really struggled with rare RNA or low-stoichiometry sites. But as this landmark 2017 Nature Methods paper highlighted, advanced techniques like Nm-seq now allow us to map these sites with absolute base precision. To uncover these deeply hidden stealth mechanisms in our own basic research, we absolutely have to be able to see them at single nucleotide resolution. So how do we actually achieve that efficiently in our daily workflow?
Which brings us perfectly to Section 4, CD Genomics Sequencing Service. This is the ultimate tool for researchers looking to map the epitranscriptome without spending months optimizing protocols.
CD Genomics offers a really comprehensive 2-O-RNA methylation detection service using deep sequencing-based approaches like Nm-seq and Ribo-seq. They deliver a parallel, transcriptome-wide evaluation of these crucial methylation patterns. What really stands out to me is their comprehensive coverage across diverse RNA species. Whether you're looking at mRNA, lncRNA, tRNA, or rRNA, they've got you covered. Plus, they tackle the absolute biggest bottleneck in modern research, data analysis. Their service is backed by highly specialized bioinformatic interpretation tailored explicitly to answer your specific research questions and turn raw reads into actual, actionable insights.
We all know a major headache for researchers is sample input requirements. But here is an incredibly vital number for your experimental design: 100. You literally only need a starting input of 100 to 300 micrograms of high-quality, DNA-free RNA to unlock these transcriptome-wide insights for your lab.
And CD Genomics streamlines the entire process. The workflow starts with targeted chemical processing, moves rapidly through high-throughput Illumina sequencing, and finishes with advanced bioinformatics. That final step is crucial. Their expert bioinformaticians handle all the complex evolutionary conservation mapping, functional motif search analysis, and differential modification profiling, and they do it over just a few weeks. They essentially extract the high-level biological meaning from all that noise, returning your original sequencing data, experimental results, and a highly detailed data analysis report that's practically ready to be dropped into your next manuscript.
So, as we wrap up this explainer, I really challenge you to look at your current transcriptomic and structural variant analyses. Ask yourself what vital epitranscriptomic data, what stealthy molecular mechanisms might already be hiding in your samples waiting to be discovered. With the right tools and partners like CD Genomics, the hidden layer of RNA biology is finally visible. Thank you so much for joining me on this deep dive into the epitranscriptome.